Have you ever heard the name Mitochondrial Eve? It sounds grand, doesn't it? It conjures images of a solitary woman standing at the dawn of humanity. Many people imagine her as the very first woman, the biblical Eve brought to life by science. Some even believe she was the only woman alive in her world, the single mother of every human who has ever lived since. This picture is powerful, but it is also completely wrong. The real story of Mitochondrial Eve is far more subtle, and in many ways, much more fascinating. It isn't a story about one woman alone. It is a story about survival, chance, and the invisible genetic thread that connects every single one of us today. The truth is, Mitochondrial Eve was a real person, but she was not alone. Our story begins not in the ancient past, but within the microscopic universe of a single human cell. Every one of the trillions of cells that make up your body is a bustling city of activity. In the center of most of these cells lies the nucleus, which acts like a central library, holding the vast blueprint of your primary DNA. This is the DNA we usually think of, the genetic code inherited from both of our parents that determines things like our eye color and height. But scattered throughout the rest of the cell, outside the nucleus, are hundreds or even thousands of other tiny structures. These are called mitochondria, and they are absolutely essential for our survival. They are the powerhouses of the cell. Think of mitochondria as tiny batteries or miniature power plants. Their main job is to take the food we eat and the air we breathe and convert them into a special type of energy molecule. This energy fuels everything you do. It powers the muscles that let you walk. It powers the neurons that let you think. It powers the beat of your heart. Without the constant work of your mitochondria, your cells would quickly run out of power and cease to function. They are the unsung heroes of our biology, working tirelessly in the background to keep the lights on in every part of our body. What makes mitochondria especially interesting for our story is that they have a secret. They contain their own separate, tiny set of genetic instructions. This is called mitochondrial DNA, or mtDNA for short. Unlike the massive library of nuclear DNA with its 3 billion letters, the mtDNA genome is incredibly small and simple. It contains only about 16,500 letters and just 37 genes. These genes are primarily dedicated to the mitochondrian's job of energy production and self-maintenance. This small, self-contained genetic code is a relic of an ancient evolutionary past, a whisper from a time when mitochondria were once free-living bacteria. This separate piece of DNA is the key that unlocks the story of Mitochondrial Eve. Because it is physically separate from the nuclear DNA, it behaves differently during reproduction. It follows its own unique and much simpler path of inheritance. While your nuclear DNA is a shuffled mix from your mother and father, your mitochondrial DNA tells a much more direct story. It comes from one parent and one parent only. Understanding this simple rule of inheritance is the first step toward tracing our ancestry back through countless generations and across millennia to a single point in the distant past. It is a detective story written in our DNA. To understand this story, we must travel back in time. When a sperm and an egg cell unite to create a new life, they both contribute genetic material. Sperm-egg cell, their union. The egg cell is packed with thousands of mitochondria. The sperm is designed for speed and delivery. It carries nuclear DNA in its head. Its few mitochondria are mostly in the tail. Those tail mitochondria are typically destroyed at fertilization. So virtually all mitochondria in a new baby come from the mother's egg. You inherited your mtDNA from your mother, your maternal grandmother, your maternal great-grandmother, and so on. It's purely matrilineal. Men have their mother's mtDNA too, but men cannot pass it on. They're a genetic dead end for that line. That maternal-only inheritance is a powerful tool for scientists. It's like a surname passed only from mother to child. It lets us trace one clear line of ancestry, without the shuffling of nuclear DNA. Nuclear DNA is tangled branches. mtDNA is one clean branch back to the source. Tiny random mutations occur, like typos. They happen at a fairly predictable rate over thousands of years. By comparing mtDNA and counting differences, scientists build a molecular clock that estimates when a common maternal ancestor lived. Armed with the knowledge of how mtDNA is inherited and how its molecular clock works, scientists embarked on a groundbreaking project in the 1980s. Alan Wilson, Rebecca Cann, Mark Stoneking. they decided to use this tool to build a global human family tree. They began collecting mtDNA samples from people around the world: Africa, Asia, Europe, Australia, the Americas. Their goal: compare all these mtDNA sequences and see how they were related, grouping similar ones and counting mutations to estimate split times. The program built a vast evolutionary tree, showing close branches and distant ones. Tracing branches back in time, a remarkable pattern emerged. All lines eventually converged. Every person in the sample shared a common maternal ancestor. That ancestor was nicknamed Mitochondrial Eve. Genetic evidence pointed to one continent as the cradle of humanity, with the deepest branches in African populations. The story of our origins was becoming clearer, written in the code of our mitochondria. So, who exactly was this woman we call Mitochondrial Eve? Based on the genetic evidence, she was an anatomically modern human, a Homo sapiens just like us. The most robust scientific estimates place her life somewhere between 150,000 and 200,000 years ago. Geographically, the data strongly indicates that she lived in Africa, likely in the eastern or southern part of the continent. We do not know her real name, what she looked like, what language she spoke, or anything about her personal life. The name Eve is simply a convenient and memorable label, chosen for its symbolic connection to a maternal origin story. The most important concept to grasp is that Mitochondrial Eve is defined by us, the people living today. She is our most recent common ancestor, specifically through the maternal line. This means if you take your mother's line, and I take my mother's line, and a person in Japan takes their mother's line, we all trace them back through time. Her mtDNA is the first one we all share. She is the woman from whom everyone on Earth today has inherited their mitochondria. Her genetic legacy, through an unbroken chain of daughters, has successfully made its way into every living human being in the 21st century. It is helpful to think of it like a lottery. Now that we understand who Mitochondrial Eve was, it is equally crucial to understand who she was not. First and foremost, she was not the first human woman. Modern humans have existed for over 300,000 years, so she lived well into our species' history. There were countless generations of women before her. She is simply the most recent woman from whom all living people descend on a purely maternal line. Second, she was absolutely not the only woman alive in her time. The effective population was likely in the tens of thousands. There were thousands of other women living alongside her, having children, passing on their own mtDNA. The key difference is that their maternal lineages have all died out over time. A woman's line becomes extinct if she has no children, or if she has only sons. Over such a vast timescale, by pure chance, all but one ancient line eventually disappear. Third, she was not our only common ancestor from that time. She is only our common ancestor for the mtDNA. In your nucleus, you carry genes from thousands of different ancestors, both men and women, who lived alongside Eve. Finally, the story of Mitochondrial Eve has nothing to do with the biblical story of Adam and Eve. It describes a mathematical coalescence point in our genetic history, not a creation event. In fact, the Adam of our Y chromosome lived at a completely different time than our Eve, proving they could not have been a couple. To put Mitochondrial Eve in her proper context, it helps to look at a simplified timeline of human history. Our story is not a short one. She appears relatively late on the scene. Around 6 to 7 million years ago, our lineage diverges from that of modern chimpanzees. Around 300,000 years ago, the earliest known fossils of Homo sapiens appear in Africa. Many Eves from this period existed, but their maternal lines did not all survive. Between 150,000 and 200,000 years ago, Mitochondrial Eve lives somewhere in Africa. She is one of thousands of women in a thriving human population spread across the continent. Her specific mtDNA lineage is the only one from her generation that survives into everyone today. Around 60 to 70,000 years ago, a small group migrates out of Africa. All non-African mtDNA types are branches of the larger African tree. By 40,000 years ago, modern humans spread across much of Eurasia. Neanderthals and Denisovans go extinct, leaving Homo sapiens as the last human species. Present day, every single one of the 8 billion people can trace their maternal lineage back to Mitochondrial Eve. Our mtDNA variations map our long migrations. The story of our genetic ancestry gets even more interesting when we look at the other side of the coin, the paternal line. Just as mtDNA is passed exclusively from mother to child, a part of our nuclear DNA called the Y chromosome is passed exclusively from father to son. Women do not have a Y chromosome. It is passed down directly without mixing. It can be used to trace a purely paternal lineage. Using this tool, scientists have identified a Y chromosomal Adam. He is the most recent man from whom all living men descend through an unbroken father-to-son line, the paternal equivalent of mitochondrial Eve. Just like Eve, he was not the first man or the only man of his time. Crucially, estimates place Y Adam at a different time than Eve, often more recent, so they were not a couple. Every gene has its own history and coalescence point. Eve and Adam are simply famous convergence points for two independent pieces of our DNA. In the end, what is the ultimate lesson? Beyond the fascinating science of genetics and the detective work of tracing our past, there lies a simple, deeply hopeful message. The discovery of a common maternal ancestor who lived in Africa hundreds of thousands of years ago is a profound reminder of our shared humanity. It is scientific proof that every person on this planet, regardless of their race, nationality, or culture, is part of the same extended family. We are all distant cousins, connected by an invisible thread of DNA that stretches back to a woman who once walked the African savanna. This idea tears down artificial barriers between us. In a world that can feel divided, genetics offers a unifying perspective. Differences in skin color, language, and customs are recent superficial developments. Beneath it all, we are built from the same fundamental blueprint. Our shared origins are written into our very cells, a constant testament to our interconnectedness. We are far more alike than we are different. Next time you look at strangers in a crowd, remember Mitochondrial Eve, a legacy of survival, incredible luck, and our beautifully complex human family.